Hello everyone, how are you all doing? This is Tithi here and welcome to another video where we are going to discuss Unit 8 important previous year questions with solutions. The topic that I'm going to cover today is one of the most important topic for CSIR NET exam, and that is 3-point test cross and microbial gene mapping. So we'll be covering both of these topics. We'll be seeing some typical type of questions which are repeatedly asked in the CSIR NET exam. Let's get started. So this first question is from 3-point test cross. So in 3-point test cross, there are two ways in which questions are asked. One way is in which they have this sample. Okay, so where they have given you the gene map and you have to calculate the number of single crossovers, double crossovers, or let's say interference or coefficient of coincidence. So those values. Another format can be where they give you those informations. Okay, they give you like these are the types of different gametes formed and what is their number, and from that information you have to find out the gene map. Okay, so for today's practice I have taken both the type of questions. Let's get started with this one first. Okay, so curled wing, ebony body color and sepia eyes are three recessive mutations that occur in fruit flies. The loci for these mutations have been mapped and they are separated by the following hypothetical map distances. Okay, so the gene map is given here. Now the interference between the genes is 0.04. The mutant CUESE fly was crossed with homozygous wild fly. The resulting F1 females were test crossed that produced 1800 progeny. What number of flies in each phenotype class is likely to be obtained in the progeny of the test cross? Okay, so first of all, this information is given to us. Okay, interference, right? So, let me write down the cross first. So we have this mutant. So CUESE homozygous, okay, is crossed with a homozygous wild type fly. Okay, and the resulting, so wild type will be what? Let's say it will, if we make it the CU plus E plus SE plus CU plus E plus SE plus. Okay, so in F1 what we will get? CU plus E plus SE plus, and the other chromosome will be CUESE. Now this is crossed with, it is test crossed, right? So that means it is crossed with the homozygous recessive parent. Okay, and then we have to calculate for the F2, what will be the result in the F1 cross. Now what information is given to us? It says that interference is equal to 0.4. Okay, now interference is what? Interference, it helps us to estimate like how many of the, or what percentage of the expected double crossovers are not happening. Okay, so that's why what happens, coefficient of coincidence, which I am representing as COC. So COC is equal to 1 minus I. Okay, so coefficient of coincidence is a measure that determines how many of the expected double crossover did happen. Okay, and so whatever is the expected double crossover that did not happen, that is because of interference. Okay, so coefficient of coincidence from this value we can easily calculate is equal to 0.6. Okay, now I will, because we don't have enough space here, let me open the whiteboard. So interference. So interference is equal to 0.4. That means coefficient of coincidence is equal to 1 minus 0.4. That is equal to 0.6. Okay. Now what is the other formula for coefficient of coincidence? So for solving these questions, definitely you have to have a good grasp on the understanding, and also you should have to remember these formulas. Okay, how to calculate interference, how to calculate coefficient of coincidence, how to calculate the recombination frequencies, all that. So what is coefficient of coincidence? Coefficient of coincidence, the other formula for that, if we have to write down, so it is observed frequency of double crossover divided by expected frequency of double crossover. Okay, so from here it will help us to calculate the number of double crossover. So why we are doing it? Because we want to calculate the number of double crossover. Okay, that is important for us to answer the question. Okay, so what will be the observed frequency of double crossover? So observed frequency will be coefficient of coincidence multiplied by expected frequency of double crossover. Okay, now coefficient of coincidence we know is 0.6. Now what is observed what will be the expected frequency? Okay, now see, for finding out the expected frequency, so we have to draw the map, is important here. So CUESE, right? Now distance between CU, will make it more accurate here. Yeah, so distance between CU and E is 20 map units and E and AC is 12 map units, right? Now, so this is in map units. So map unit is equal to the percentage recombination frequency, right? So what is the chance of recombination between CU and E? So recombination frequency between CU and E will be, like, if you take out the percentage, it will become 0.2. And the recombination frequency between E and SE will be 0.12. Okay, now since this recombination frequencies are are independent events here, like if we consider them independent, then only we will be able to calculate the expected frequency of double crossover. So expected frequencies, how do we calculate? It is the multiplication of the individual recombination frequencies. Okay, because that much is our expectation. Now it does not match with the expectation because of interference. Okay, simple. So it will be, so what is the expected frequency of double crossover? It is 0.2 multiplied by 0.12. Okay, so this will help us to calculate the observed frequency of double crossover. So when you do that, you will get the value of 0.0144. Okay, now this is the observed frequency. We have to calculate the number. Okay, so number of observed double crossover will be how much? Whatever is its frequency multiplied by the total progeny. Now how much was the total progeny? This number is given in the question as 1800. So we have to multiply this frequency with 1800 and that will give us the number of double crossover. So when you do that, it will come as 25.9 something. So which we can approximately write as 26. Alright. So one part we have done. Number of observed double crossover is 26. Okay. So let's see here. So which all the options are saying that number of double crossover as 26. So you can eliminate option B. You can eliminate option D. We are left with A and C because both have double crossover as 26. Okay. So we have already done 50-50. Let's move ahead and see. So what else we have to calculate? So we have to calculate the single crossover products, right? So between each of them. Okay. So what will be the, so the recombination frequency if we write down. So I'm drawing this map again. And this is 12 map unit. Okay. Now the recombination frequency between CU and E. Okay. How much it is? We know that it is 20 map units. Okay. Or we can write it as 0.2. Right. Now what will be this recombination frequency? This recombination frequency will be the total recombinants divided by total progeny. Okay. Now when we are saying total recombinants, so total recombinants between CU and E divided by total progeny. Okay. Now, what is total recombinant? So, in total recombinant, we have to calculate the single crossover products plus the double crossover products, because in double crossover, one recombination event happened here as well, right? So, double SCO plus DCO divided by total progeny, which is 1800. Okay. Now, in this, we know the number for all, we have to calculate the SCO. Okay. So, SCO plus how much is DCO? 26 divided by 1800. Okay. So, when you solve it, move it to the other side and solve. So, then SCO will become, so you have to move this to this side, and then so 0.2 multiplied by 1800 will come as 360 minus 26, it will come as 334. So single crossover between CU and E is 334. Okay. What about the other one? Same way, recombination frequency between E and SE. Okay. So here also it will be the single crossover product between E and SE plus double crossover, because in double crossover one crossing over is happening there as well, and divided by 1800. Okay. So this is 0.12 SCO plus 26 divided by 18. Okay. So SCO between E and SE will be equal to, so 0.12 multiplied by 1800 will become 216 minus 26. So it will be 190. Okay. So 334 and 190. Now let's see our options. So you can see option A, the single crossover between CU and E is 334. Single crossover between E and SE is 190. Okay. So from here only we are sure that A is the answer, but let's calculate for the non-recombinant also. So what will be the non-recombinant? So non-recombinant will be total progeny minus recombinants. Okay. So how much is the total progeny? 1800 minus what is the total recombinant? So recombinant is 334 plus 190 plus 26. Okay. So when you subtract them all, it will become 1250. So answer is option 1. Okay. Now moving on, as I told you that this is the second type of question which can be asked, where they give you the type of all the gametes which has been formed, all the individuals which has been formed as a result of 3-point test cross, and they ask you to find out the distance, or they ask you to construct the gene map. Okay. Now in that case, how do we do? So here the first step usually is to find out the middle gene, but we don't have to find out because it's already given that B is the middle allele. So it is just saying that find out the distance between AB and BC. Okay, that means B is in the middle. Okay, this is the order. Okay, order is given to us. All right. So again, let's write down the cross for our better understanding. So this, that is, so I'm going to write down in the linked gene format, okay, because then it will be easy for us to understand. So we are getting a F1, which is one chromosome is all dominant, one chromosome is all recessive. Okay. Now when that is done, our test cross, okay, and this is the next generation that we are getting. Okay. Now in here you see the genotypes are given in the normal format. So if you have to convert, so we have to convert it in the linked gene format. So in that case, what we have to remember is that this recessive parent, the homozygous recessive parent, is giving small a, small b, small c, right? So from everywhere, small a, small b, small c will come on one chromosome. Whatever is the other one left will come on the other chromosome. Okay. So for example, if this genotype, I have to write it in linked gene format. So it will be capital. So one chromosome will have small a, small b, small c. Take it out. What is left? Capital A, capital B, capital C is on the other chromosome. Okay. Now, what are the double crossovers? So we know that from this table, whichever class has the least frequency will be the double crossover. So these two varieties are the DCOs. Okay. Now we have to figure out the SCO between each pair. So let's consider A and B first. Okay, so if this genotype, so if this is the genotype of the heterozygote, so in that case, if crossing over is happening here, okay, so what will be the SCO produced? SCO will be capital A, small b, small c, and small a, capital B, capital C, right? So what all classes are this one? So remember, in all of them, small a, small b, small c will go to the other chromosome. Okay, so this is one of the SCO between A and B. Okay, where you take out small a, small b, small c, you are left with small a, capital B, capital C, right? This variety. And the other one will be this one, where you again take out small a, small b, small c. So you are left with capital A, small b, small c. Okay, so this is the other SCO between A and B. Okay, so now if we have to calculate the distance between A and B, okay, so the formula is very simple. Recombination frequency between A and B. So it will be the total recombinants divided by total progeny. So total recombinant will be the SCOs between A and B plus DCO divided by total progeny. Now when we add that up, so it will come to 1000. Okay, whatever classes are given, when we add that up, so it comes to 1000. Now what is SCO? So one class was 100, one was 120, and for the DCO it was 14 and 16, right? And then when we divide it with 1000, so and we have to multiply it with 100, okay, because recombination frequency, the map unit, the distance between AB is given as recombination frequency percentage. Okay, so we have to multiply all of that with 100. Okay, so when you solve this, what you will get as 25 centimorgan or map unit. Okay, now similarly we will do for B and C. So for that, let's check out. So definitely that means the class with the highest number are the parental types. So the other two varieties are this, right? So these two are the SCOs between B and C, and you can check also that in case suppose recombination is happening here, then what all classes will you get? I'm just erasing it to avoid confusion. Okay, so what all classes will you get? Capital A, capital B, small c, and small a, small b, capital C, right? So these are the two classes. So again, what will be the distance? Distance between B and C. So it will be again that single crossover. So single crossovers here are 65 plus 75 and plus the double crossovers. We cannot forget the double crossovers, because in double crossover, one recombination event happens at between each pair of genes. Okay, and whole multiplied by 100. So that will give us 17 map units or 17 centimorgan. Okay, so the answer here will be option C. Very easy, you just have to remember the concept and few formulas. Now next question, moving on to microbial gene mapping. Now this is the type of question which is very frequently asked from this conjugation experiment, interrupted mating experiment. So using interrupted mating, 4 HFR strains were analyzed for the sequence in which they transmitted a number of different genes to a F- strain. Each HFR strain was found to transmit its gene in a unique order as summarized in the table. Okay, so we have to analyze this table and make the gene map. So we will start with the circle. Okay, now you see each of the strain one by one. So starting with strain 1. So strain 1, first A is going. Okay, so they will be placed in the same order in which they are going. That is how we will construct the gene map. So if I am placing A here, that means next comes B, then D, then F, and then H. Okay, now because A is going first followed by B, that means the integration site, here you see the arrowheads are mentioned, so that is the integration site of the F plasmid, so that also we have to represent. So it has to come here for 1. Okay, now what about for 2? 2 BAL. Now we have to look for the overlaps. Okay, so if BA is on this side, that means LMK will be the other side of B. Okay, now here B is going first followed by A, that means the integration site of F plasmid is here, and the arrowhead will be pointing towards D. Okay, because at the head of the arrow there will be cut and the tail will be joined with the DNA, which will be taken to the recipient. Now what about strain C? So it is MK. So again we have to look for overlap. So we have got an overlap here of MK. So it is MK, then J, then G, and H is already placed. Okay, now M is going first followed by K. So here has to be the integration site for the third strain, and for fourth strain F, H, G, J, K. So all the genes are placed. We just have to place the integration site of the F1 plasmid. So of the fourth strain, which will be here. Okay, before F. So F will be at the tail side. Alright. So once we have drawn this kind of a map, match it with the options that are given. This is option 1. This is option 3. So when we check it, what do we get? That option 3 is the answer. Okay, so the gene map that we are getting here is exactly what we have drawn. Okay, so here you have to take care of, like when you are comparing your answer with the options that is given, you have to take care what is the relative position of all the gene markers, and also you have to take care about the positioning of the F plasmid. Okay, so for one strain it has to be between L and A, pointing towards A. For the second strain it has to be between B and D, pointing towards D. Okay, so these are the things you have to compare. Now this is also a type of question which has been asked multiple times, which is a relatively easy question. Just you have to focus on the concept. So in a conjugation experiment between bacterial HFR strain X and F- cell, lac gene enters the recipient in 4 minutes, but the F- cells remain auxotropic for Leu, Trip, URA, Glu, Phi, and Gly. The mating is then allowed to proceed for 20 minutes and lac plus X conjugates are selected. Of the lac plus cells, so this is the percentage in the recipient that are mentioned, okay, for the different markers. Select the correct order of the gene. Okay. Now here what concept we have to apply? So in case of interrupted mating experiment, so what happens is when the HFR cell is conjugating with the F- cell, so the physical contact is necessary. Right now, sometimes what happens if the genes are placed very far apart, so lot of time will be taken for the transfer, and so when lot of time has to be taken for the transfer, means for lot of time those two cells have to remain in physical contact, which becomes difficult for the bacterial cells, okay, because they are constantly in the Brownian motion. So because of which, what do we see? That the genes which are placed very far apart from each other, so the chances of the recipient cell receiving both the genes decreases. Okay, so here if we have to consider that lac is going first, okay, it is going for at 4 minutes. Okay, so and we are selecting all the lacs plus X conjugates, means all the cells who have got lac we are selecting. Okay, then in which order the genes will be passed? So basically the cells will receive, maximum of the cells will receive that gene which is present right next to lac. Okay, so lac is going first for sure. You can write it down here. Now after lac, what will be going? So the recipient cell will receive the next highest, the maximum number of cells will receive the next gene which is present right next to lac. Okay, so what is that gene? So 98% of the cells are receiving trip plus, means right next to lac it has to be trip. Okay, now similarly we will keep on going to the for the genes in the decreasing order of the percentage. Okay, so after that there is gly, means next in the line is gly. Okay, so do you all understand why this number of recipient, the percentage of recipient cell is decreasing? Because farther the genes are placed, more is the chance of the conjugation to get interrupted naturally. Okay, and if it gets interrupted, then the gene will not be passed. Okay, so the order here will be lac, trip, gly. Then after gly it will be glu, because 65. Then it will be lu plus. Okay, and then finally, then ura, and then phi. Okay, so you can see it is so easy. It is just like arranging them in this in the decreasing order. Okay, but for that you have to have the understanding why we are doing that. Okay, so farther the genes are placed, lesser will be the number of cells receiving it, because more time will take for the transfer, and between that time the chances of physical contact to interrupt spontaneously is high. Okay, so answer will be option D. All right, so with this I'm going to stop the video. I hope you found all the questions interesting and now you understood the basic concept, and you will try more questions, and please let me know whether you are able to solve them or not. Thank you everyone. Have a great day.